Hey everyone, today we will be presenting on the conservation of the Malayan tiger, antara characteristic seni and the molecular technique that can contribute to its long-term survival. The Malayan tiger is a critically endangered subspecies found only in peninsular Malaysia. Over the past several decades, its population has declined dramatically from around 3,000 individuals in the 1950 to approximately 200 individuals in 2018. Factors have contributed to this population crash. Between 2008 and 2020, Malaysia records 652 human tiger conflict cases and an estimated 39 poaching incidents involving tigers. Additional threats include habitat fragmentation, declining prey population, disease transmission, and roadkill incidents. As the population continues to decrease, effective conservation strategies are becoming increasingly important. In addition to traditional conservation methods, molecular techniques have emerged as valuable tools for wildlife management and species preservation. The objective of the study are to examine the conservation status of the Malayan tiger and explore three molecular techniques used in its conservation. These techniques include DNA barcoding for species verification, microsatellite analysis for individual profiling and population monitoring, and biobanking with cryopreservation for the long-term preservation of genetic resources. Through these approaches, conservationists can obtain accurate genetic information, monitor population health, and preserve valuable genetic materials for future conservation and breeding programs. Overall, we expect this molecular technique to contribute to more effective conservation planning and support the long-term survival of the Malayan tiger in Malaysia.
Nurdin opened the conservation overview. My part focuses on our first in situ molecular tool, DNA barcoding. So what is DNA barcoding? Just like a supermarket scanner reading a unique black and white barcode to identify a product. Similarly, DNA barcoding utilizes a short standardized genetic sequence from a specific mitochondrial gene to rapidly resolve taxonomic uncertainty and accurately identify a biological species. The main goal is to use genetic data to find Malayan tiger even when we cannot see them. Now, how does this work in conserving the endangered wild Malayan tiger? Tracking tigers physically in that tropical rainforest is incredibly difficult. Therefore, researchers utilize non-invasive genetic sampling by collecting field samples like poop, fish catch mark, and environmental DNA from the ground without disturbing or catching the animal. Once DNA is extracted, we perform polymerase chain reaction, amplification, and sequencing targeting a specific mitochondrial gene, which is the cytochrome B gene. This gene acts as a genetic barcode. In the study by Alberry et al. 2024, this exact technique was used to scan 295 collected cannibal samples and peninsular Malaysian forest complexes. Why is this species verification so critical for conservation? In the wild, tigers can look identical to other co-predators, such as leopard or black panthers. Without DNA barcoding, this identification leads to inaccurate population data. By looking at the cytochrome barcode, researchers successfully fit out 9 tiger species and verify exactly 26 high-quality samples belonging to Panthera tigris taxonomy. This precise verification ensures that valuable funding, tracking, and protection efforts are applied to ensure tiger habitats. Furthermore, validating the right species is the crucial foundation to proceed with microsatellite analysis for individual identification, which will be elaborated by our next presenter.
Next, researchers use microsatellite analysis to identify individual tigers through DNA obtained from non-invasive samples. Firstly, researchers collect non-invasive samples such as tiger scut from the forest without disturbing the animal. After that, DNA is extracted from the sample in the laboratory to obtain genetic material. The DNA then undergoes PCR amplification, which helps increase the amount of DNA so it can be analyzed more clearly. Next, microsatellite analysis is carried out by examining specific repeated DNA sequences that are unique to each tiger. Since every tiger has a different genetic profile, researchers can identify individual tigers accurately. Finally, this information is used for population monitoring, including estimating popular size, tracking movement, and assessing genetic diversity for conservation purposes. After understanding how microsatellite analysis works, we can now look at its conservation applications in tiger conservation. Okay, firstly, microsatellite analysis helps in population size estimation, where researchers can estimate the number of tigers in a particular area by identifying different individuals from DNA samples. Secondly, it is used for genetic diversity assessment to evaluate the level of genetic variation within tiger populations, which is important to reduce the risk of inbreeding. In addition, microsatellite analysis also supports relatedness analysis, allowing researchers to determine family relationship and breeding patterns among tigers. Furthermore, the data obtained can contribute to conservation planning by helping authorities design better strategies for habitat protection and species management. Okay, lastly, microsatellite analysis can support anti-poaching efforts because DNA evidence can be used to identify illegally hunted tigers and track wildlife crimes. Therefore, microsatellite analysis is a valuable molecular tool for protecting endangered tiger populations.
On previous molecular technique discussed by my group members, I will now explain another important aspect to conservation approach known as biobanking and cryopreservation for the conservation of the critically endangered Malayan tiger, also called as Panthera tigris jacksoni. One approach is biobanking, which is involved collecting and storing biological material such as blood, tissue sample, DNA, sperm, oocyte, and embryos from individual tigers. These samples are stored in specialized facilities known as genome research bank, where they can be preserved and assessed for future research and conservation program. To ensure long-term storage, cryopreservation is used. This technique preserves biological materials at ultra-low temperature, typically in liquid nitrogen at minus 196 degrees Celsius. At this temperature, cellular activities are effectively stopped, allowing the genetic materials to remain viable for many years without significant degradation. The importance of this technique lies in its ability to safeguard genetic diversity. Store sample can later be used in assisted reproductive technologies such as artificial insemination, in vitro fertilization, and embryo transfer. This application can support captive breeding program and future genetic rescue initiative aimed at strengthening population viability. Furthermore, conservation biobank provide a long-term safeguard against the permanent loss of unique genetic trait. Wildlife biobank are becoming increasingly important for preserving genetic research and supporting future conservation strategies.
In conclusion, molecular data has shown that landscape fragmentation has severely reduced the genetic variation of the Malayan tiger, making it one of the least genetically diverse tiger population in the world. This situation is alarming because low genetic diversity can increase the risk of inbreeding, reduce adaptability, and threaten the long-term survival of the species. To address the issue, the first recommendation is to implement genetic profiling for all the captive Malayan tigers in zoos and rescue centers using the 16 microsatellite local method. This approach can help conservationists identify genetic relationship between the individuals, allowing more effective selective breeding programs and reducing the risk of accidental inbreeding in captivity. Secondly, local and indigenous communities should be further empowered in conservation efforts. For example, the orang asli communities, particularly the Jahat tribe in Royal Belum, have demonstrated their activeness as community rangers. By combining their traditional knowledge and tracing skills with conservation initiative, they can help detect and remove snares, monitor wildlife, and deter poaching activities while also benefiting from sustainable employment opportunities. Lastly, patrol efforts and monitoring systems should be expanded across key tiger habitats, especially in the Belum Temenggu Forest Complex and the Central Forest Fund. Increasing the number of field patrols and camera traps can improve the detection of poaching activities and provide valuable data on tiger population and their movement. By combining genetic management, community involvement, and stronger protection measures, we can improve the chance of ensuring the long-term survival of the Malayan tiger for future generations. Thank you.